Hello, everyone. I think we'll wait a couple minutes just to let some more people log on. I mistakenly uh, sent people the wrong link in my first email, but I resent the correct link. So hopefully everyone will eventually be able to get on. But it looks like we've got, what, 22 people here already, so that's great. So normally we hold this meeting in person in conjunction with the Idaho chapter of the Wildlife Society meetings. I suspect we may actually get more people participating this way than we normally do. Here is just a brief outline of what we're going to be trying to do. We can keep this pretty loose. Hopefully we won't have any technical problems. I think normally we start out with introductions. We'll probably not do too much of that, at least initially, because it could take a while to turn everybody off and on and get through. We're up to 26 people now, so that probably would take a lot of time. But we'll during the break, I'll try to set up a Mentimeter quiz or poll, and you can sign in there so we'll know who's attended the meeting. We'll go over the national and regional park updates. Laura Sprague, who's with the U.S. Fish and Wildlife Service, is the new Northwest Park chair. And she's going to give us an update on national and regional park. There isn't much in the way of her presentations at the meeting, well, at the 2021 meeting, but I'll mention those talks. And then we'll do statewide herp news, and we've got 4 different people presenting about that. And then we'll have a break, I think. We'll see how the timing goes. And then we'll go around the regions, and then that'll be kind of roundtable. So we've got several people that will talk about what's going on in their region, and then that's an opportunity for anyone to chip in with what they know is going on in their region. So during the break, we'll have you sign in. In my teaching, I've been using something called Mentimeter, and it's worked pretty well for that. So I thought we'd give that a try during the break and see if we could document who was attending the meeting. My name is Chuck Peterson. I'm a professor in the Department of Biological Sciences at Idaho State University. Okay, so our first person to present is Laura Sprague from the U.S. Fish and Wildlife Service. And I think I've got the right slides in place here. So if you want to unmute yourself, Laura, and then I'll just advance the slides when you ask me to.
Perfect. Okay, so as Chuck said, I'm Laura Sprague. I am an aquatic animal pathologist, mostly in fish, with dealing with fish, but I do a few amphibians as well along the way. So currently I am the senior co-chair of Northwest Park, and right now the only co-chair. So I'm glad to be here with you. I'm based out of Orofino, Idaho. Next slide. So just for those of you who don't know, Northwest Park area is pretty large. It encompasses several states and 3 territories. And so there's a little bit of overlap between those, but we're a pretty big group of people. Most of our members are from Washington, Oregon, and Idaho, and a few from BC, and a few stragglers here and there. If you know anybody in Montana, Wyoming, that is wanting to be a part of PARC or Northwest PARC, encourage them to sign up so we have some representation from some of those areas. Okay, next slide, please. Okay, so the steering committee asked me just to mention this briefly. This was prepared by Dee Olson and David Pilliard. So these are the main threats to our amphibians and reptiles nationally, and these are the ones that we're going to focus on within Northwest PARC over the coming years. And so there is some funding opportunities that are going to be associated with research that's being done with habitat changes for both amphibians and reptiles. That includes roads, hydrologic changes, mining, energy development, rural development, agricultural practices. The big moneymaker right now is invasive species, which can alter communities of native species, of course. And there's a significant amount of money going into bullfrog eradication right now. So if anybody has any projects that are or any interest in that, let me know. Of course, climate change is a big issue, especially with the new administration, chemical contamination, and then of course, illegal trafficking is also a big issue. Next slide. So as I said, we have a new senior co-chair, that is me. We have 5 new steering committee members, all energetic and ready to work. If any of you are interested in being a part of the steering committee or helping out with some of the activities that we have going on, I encourage you to get a hold of Chuck or myself, or register on the PARC website for Northwest PARC, and just let people know that you're interested. We're always looking for fresh ideas and new innovations and things. So we're going to be working on updating our website. It's a little bit behind. Unfortunately, SNDV, which is the Society for Northwest Vertebrates, we usually have our meeting joint with them. In fact, Northwest PARC is kind of actually started as a subcommittee of SNDV, and then kind of branched out to what it is now. And there's been some leadership failures throughout both of those organizations. And so we're working on trying to rebuild it in the Northwest. So our website hasn't been updated since 2018. We're going to be working on that. We're probably going to be reaching out to folks, trying to get some articles of research that's going on in the area. So if you have some cool research and you want to be a part of that, we can spotlight you on our website or in our quarterly newsletter. Or if we'll be spotlighting people as well and reaching out to folks. We also have what you call the In Your Area initiative.
Please take her outside instead of getting her all riled up. I'm on a meeting.
So with the In Your Area initiative, it's basically a research, there's not a lot of communication between areas. And so this was originally designed for folks to be able to communicate. And so if you're working on a specific project and you want to know if there's other folks within the native range of a particular species that you're working with that are also working on it, maybe across state lines or in another region within the state, then we encourage people to actually go in, talk about the research project briefly that you're doing, and it's a way for folks to be able to communicate. It's been a little bit dead in the water as well. I think there was like 6 or 7 projects that were listed in there, but we're really going to try to push that for communication purposes, and just so that we don't, waste our time and energy, replicating a project that maybe we can, use our funding or our resources a little bit better to get a bigger, broader picture of things. As I mentioned a little bit earlier, we're working on a new quarterly newsletter. It used to come out quarterly, so we're going to reinitiate that. I mentioned spotlighting on the website. We're looking at holding a virtual meeting in the near future, but we haven't determined what kind of format that's going to be. There's some discussion on whether we're going to pre-record it and have those presentations available, and then have a roundtable or a panel type discussion at the end of the month on a given day to talk about, to be able to ask the presenters questions, or just have basic discussions on some of the things that they learned in those pre-recorded meetings. We're also trying to determine whether we're going to run it over multiple days. So maybe like every Monday at 2 o'clock for 6 weeks, or if we're going to run it over a weekend. I know Southwest PARC just had a meeting last weekend that ran Friday and Saturday with 0 breaks. And it seemed to be pretty successful. They had over 300 people register for that meeting. So we might look at something like that. If any of you folks have would like to be included in that virtual meeting and maybe potentially give a presentation, that would be amazing. So let Chuck or myself know that you're interested in presenting, because we'll be reaching out, and the number of presenters that we get will probably determine if we're going to do multiple days, or if we're going to, help determine the format that we're going to pursue. We still have some folks are wanting to have a meeting such as this where it's live, but unfortunately, even though it's easier to attend a virtual meeting sometimes, there's a lot of conflicts with regular work duties, and so it's not as easy to sit down for a time slot for some folks. So if you have some ideas of how you think that that would be best presented, or if you were interested in attending, how you would prefer that to go, please give us your feedback. And then I'm going to just wanted to briefly mention that there's a new data tracking tool being used in BC for herp conservation, and Leanne is willing to share that with folks. So if you're looking for a new tool to be able to do that, again, let us know, and I can put you in touch with the right people to get that information. Next slide, please. Okay, so updates on National Park. Park has a 2-hour workshop this week at the North American AFWA meeting, or I'm sorry, not this week, the second week of March. That's March 5th through 8th, 2021. There's gonna be an hour devoted specifically to BSAL, and what we found, what the BSAL task force is working on, and just some basic information. And then there's going to be another hour presentation specifically for turtle trafficking, which is a really big thing back east right now.
Laura, you might want to tell them a little bit about what BSAL is.
Okay, I can't pronounce it. So BSAL is the counterpart to BD. We do have BD here in Idaho in the Pacific Northwest. So BSAL was found in 2015, was discovered in 2015 in Asia, and that caused, when it was found, it was thought to only be causing mortality in salamanders. And so there were some studies done. It caused a change in the Lacey Act, and the first interim rule to be produced by U.S. Fish and Wildlife Service, prohibiting the importation of 201 salamanders in 2016. It was the first time that we've had to make an interim rule prohibiting the importation of animals in several years, almost 20 something years. And so with that, the BSAL task force came into play. So the BSAL task force is actually a preventative task force. There's several subcommittees on that. There's several types of agencies representative, and it's the first time that the pet trade industry actually had a vested interest in trying to prohibit the importation of disease. Unfortunately, with the Lacey Act only prohibits the importation of wildlife and fish. animals, but it doesn't, there are no regulations for the importation of disease. And so we can't make anybody do any kind of disease testing. And so the United States actually has the second largest diversity in salamanders in the world, with the most diverse area being, of course, the Appalachian Mountain Range on the East Coast, and the second largest diversity in the U.S. is the Cascade Range in Washington. And so with Northwest PARC, it's a very real concern if BSAL is introduced into the United States. To date, there has been zero findings of BSAL. There's been some close calls. Like I said, we've stopped the importation of animals, and they've been, several states have regulations in place that actually, before animals can be brought into pet stores, they, like New York, they will actually do the testing for some of those. Other states aren't on board with that, but I guess there's a lot of concern because there were some dying salamanders in New York, and so they put that in place. So some of the things that we work on the BSAL task force, we actually have a diagnostics team that actually does ring testing. So there's 40 labs that are involved in that, nationally and internationally, including Canada, and I guess they're all in North America because it's Canada and Mexico. But with that testing, so we send out ring test samples, positive controls, and then have everybody run those samples to make sure that their equipment and the reagents that they're using will detect BSAL at different levels, so that if BSAL is ever found, that we actually have the ability to send to multiple different labs. We have a research group, and so they talk about the needs of research as far as what kind of species are going to be susceptible. There's some susceptibility studies going on with Jonah Piovia Scott's group out of Vancouver, Washington at the university there. There's other studies going on other places. We also have a management group that actually is quick response and management, so that if a manager, a lot of times, there's a lot of red tape and paperwork, and there's a lot of regulation that you have to sift through, and so there's some step-by-step procedures that have been developed, so that if BSAL is detected, then a manager of that particular unit knows the steps and processes they need to go through in order to be able to contain the BSAL or eradicate it from that particular area. And then there's also some other normal groups that like outreach. So there's some websites, and there's some articles that come out, and there's some white papers that come out. There's a disease task team as well. So anyway, there's a lot of stuff going on in the background, and that is a still pretty active group, considering that they started in 2015 and have come a long way with that. There is a national strategic plan that is set out by the group. That's one of the things that they're tasked to do, and so you can find that on salamander.org website. Any questions about that, go ahead and get a hold of me. I'll give you my email here at the end. So the Joint National Subcommittee, JNSC, is for National Park. That includes state representatives. It's all of the co-chairs of the different regions, as well as some other folks. Fish and Wildlife Service, state representatives are on there, and then there's a USGS representative as well as a Forest Service representative. And with that voting committee, they would like to increase state chapter involvement with PARC in general. And so Idaho, you guys actually have a group. I think there's only 7 states that have individual chapters. Chuck is really good about giving us information about what's going on within the chapter and accomplishments and field trips that you guys go on. And so we're one of the active groups, the Idaho group. But they want to invite state chapter leaders to come in, or state chapter members to come in and at least give them some updates or some cool information, just so that you have a better idea of what goes on at those meetings, and that they can be more aware of what goes on in the field, because a lot of them don't get out in the field anymore. So they're working on filling some vacant positions. There's a junior co-chair for the national park. They're looking for an industry rep and a non-governmental rep, as well as an AFWA state coordinator rep that has been vacant for 3 or 4 years, three years now. But that position and these are all, so the AFWA state position and the junior co-chair, I think, are paid positions. The junior co-chair is not. The industry rep position is a paid position, and the AFWA state coordinator position is a paid position. And they're not only dealing with herps, but also aquatic invasive species. And I think that's it for National Park. So we also have a disease task team that I wanted to mention, because a lot of folks don't know that that's out there. And this is one of the teams that falls under the national park. So there's a website right there that you can go and check this out and get more information. But we do, there's a new, what we call HADIS, the Herpetofaunal Disease Alert System. And so basically, if you find any diseased animals, you take a picture. There's some questions on the website that they'll ask you, like, where did you find it? What time of day? Was it a mortality, or was it just exhibiting morbidity? And then you email that picture and that information to herp_disease_alert@parkplace.org. There's 3 folks, veterinarians, that will answer that email and give you, get back in touch with you if they have more questions. And then that will also, they'll be in contact with a state representative or state veterinarian that would be in charge of gathering more samples if it was a disease of concern. And then they'll be back in touch and give you the answer of what they think it is, or they might be in touch with you to try to grab another sample, if possible, depending on whether you found it recreationally or if it's part of your job duties. So I just want everybody to be aware that that's out there. There's a lot of folks that don't know that that is out there. It's been pretty successful so far. It's been highly publicized on the East Coast, and so we've gotten most of our contacts from that way, but just know it's there. So the disease task team is also producing a lot of fact sheets on several different diseases, including snake funnel disease, BD, BSAL, rhinoviruses, just about anything that we have information on. And you can find those brochures and informational leaflets at that website that's listed over there to the left. So non-game wildlife is an emerging topic nationally, just because of the interim rule that was passed in 2006 on BSAL. And there's a lot of other things that are going on. Unfortunately, as we all know, there's not a lot of money dedicated to non-game wildlife health. So with that being said, there are funding opportunities that are going to be coming up. And the reason for that is because they've been able to piggyback with the Invasive Species Initiative. And so this week is actually National Invasive Species Week. And there are a lot of non-game wildlife that are invasive species, which include amphibians and reptiles. And so they've been able to piggyback with that. And so there's research money available for non-game wildlife that is that may be impacted by the invasive species. And so that's how we are going to get funding for that. And then just to let you know that there is a paper on biosecurity They're calling it the dirty, working in a dirty to clean environment, which is in press. It will be in the March issue of Herp Review that was written by Katie Heyman and Dee Dee Olson, and a few others that are on that. And basically, you know, everybody knows how to plan a field season. Everybody knows how to disinfect when you're at a site. But this is in a situation where you didn't know a site was contaminated. And so you're at a site early in the day or early in the week in which you're at a facility that, or at a location in which you know is contaminated after you get there, and how to disinfect and how to go about moving to a cleaner area. And so it's got some really good information on how to do that. Next slide. So these are just some upcoming events to mark your calendars with. Of course, this is National Invasive Species Week. And so a lot of the state coordinators have put together invasive species presentations throughout this week. So Washington has several, if you want to learn about murder hornets and other things that are non-herp related, they have an array of stuff that they're talking about. Idaho has a couple this week, and then Oregon has a week full of presentations as well. I think most of the presentations are during the lunch hour. The Southeast PARC annual meeting is coming up it's being held this week as well. Idaho chapter AFS meeting is next week. There are going to be a couple of talks on amphibians, I hear, but I have not seen them at this point in time. The North American AFWA meeting is going to be virtual. It's March 5th through 8th. That is the one that's going to have the breakout session for the VSAL talk and the turtle trafficking. Normally, if you attend the North American AFWA meeting in person, there is no charge if you come as a PARC attendee, because those meetings are joint. The PARC meeting is joint with that. And unfortunately, if you want to attend that, you're going to have to pay the $150 registration fee, even if you only come for the 2 hours. So that's one of the drawbacks to that one. National Amphibian Week is, I don't have the exact date, but that's every year in May, usually the first week in May. They were talking about changing it to the week of the 25th. But I'm not sure what that is, so that's just coming up in May. The Northeast PARC annual meeting is August 10th through 13th. It's going to be virtual, and that is going to be free of charge, I believe. So the Society for Icks and Herps is going to be in Arizona, July 21st and 25th of 2021, and then they're going to move a little bit closer, and they're going to be in Spokane, Washington. They are trying to put together a live animal hands-on display for folks to be able to see native amphibians. However, there are some hoops that they're going to have to jump through. The last time they did that was in 1997. Of course, regulations have changed in Washington and Idaho since then, and so that may not be possible. But there are some set 4 or 5 different field trips in which they're going to try to get out and see some native icks and herps. Midwest PARC is going to hold a meeting, and then Southwest PARC is going to be February 22. And like I said, ours is going to be coming up, and I don't have a date for that either. And I think that is all of the updates.
Okay, well, thank you very much, Laura. That's more information than we usually, either David or I usually cover this, and we don't know as much about these things as you do. So thanks very much.
You're welcome. I told you I couldn't talk for 15 minutes, and I think I went over.
That's okay. Okay, I just looked over the schedule for the meetings, and the only true herpetology talk that I know of that's going on at this meeting is by Christina Parker, and that's going to be on at Wednesday. So my understanding is that these meetings or these talks are pre-recorded, and then later in the day there's going to be a panel. to take questions and answers and discuss things. But Christina's here and she's going to tell us a little bit about these things and some other things going on as well. I have a paper that I'll be giving that's going to be on using crowdsourced data. It's not specifically on herps, but there is some herp content in there a little bit. And then one of my undergraduate students, she just actually finished last semester, but she's talking about giving a poster on using community science to document biodiversity in a natural area here in Pocatello. Okay, so here are our, for the statewide talks. I broke this up into statewide talks and regional talks. And Charlie is actually going to talk about things in 2 different regions. So I threw him into the state. Now, Rita Dixon is supposed to present on the State Wildlife Action Plan update, but I got an email from her that she was going to be late to the meeting. So I think we'll jump ahead to Charlie, who sent me some slides that I have here. And so if you're ready, Charlie, you can present now.
Yep.
Okay. Well, there we go.
All right, so I just threw a couple slides together of some herp related enforcement stuff we've been working on over the last, I guess all this was in 2020. The biggest one that's still working through the courts is this one here in the Boise area. We had a guy that, a kid, he 20s. We had been telling him, Dr. Drew and I, for the last couple of years that he couldn't import venomous reptiles without an import permit. And so he decided he was just going to do it himself. And then I got wind of it and was able to finally actually get a search warrant, went to the house. And we found 17 non-native cobras, vipers, a whole bunch of stuff. Actually kind of scary. He had spitting cobras. We were wearing face masks or shields, and you'll see in the next picture, we wrapped the cages in plastic so that they couldn't do anything to us. But he also had 17 Western rattlesnakes, a couple gopher snakes, scorpions, and then this guy loves all kinds of critters, and I don't know how many black widows he had, but we took the scorpions and left him with the black widows. And he's still in the court process. We have, I believe, 24 charges on him, and we're still trying to find him. He moved. And so hopefully we can find him one of these days soon. Next slide. So the really pretty pictures, those 3 on the right, John Cassell came and took some photos of the evidence snakes, and I just threw some of his pictures in there, so I got to give him credit. That's why they look so pretty. But you'll see those other big tanks have plastic over them, and those were some of those spitting cobras.
Oh, entertaining. I forgot my whole.
So we still have all of those snakes. They're being cared for.
Oh.
And we're going to eventually here soon ask the court if we can dispose of them so that we don't have to take care of them. There's no antivenom anywhere in Idaho, obviously, for these kinds of critters. Next slide.
Oh man.
Betsy, put your mute on.
Absolutely. I'm so glad that campus was at least partially.
This other one is from the Boise area. Was mostly really all just about wild caught stuff. he had Western rattlesnakes, gopher snakes. Again, he's got scorpions.
How was your day?
The two people, these two cases, the guys knew each other. I wouldn't call them friends, but they knew each other. Next slide. So we actually got to this guy because Garden City Police was put him on a mental hold. He had been off his meds and was threatening to commit suicide, and then they knew that he had these snakes, and so they wanted us to come help him. So we actually went to help Garden City Police. And these pictures kind of tell you what kind of mindset he had, but also what he still has. He's not afraid to free handle these rattlesnakes. Yeah, enough said. He would exercise his rattlesnakes in the backyard. So they're in the lawn. Yeah, he'd take them out to get them some environmental enrichment. Next slide. And then Idaho Falls officers called me to help them with a case over there. They had a guy that had been around for quite a while and had kind of set himself up as a reptile rescue. He didn't have any permits, wasn't in touch with Fish and Game, but he was taking stuff in that should not have been imported into Idaho, at least without a permit. And so he had a 5.5-foot alligator. He got that as a rescue when it was a little baby. So he'd have that for quite a few years. Don't remember how long he said, 7 or 8 years. He had these 2 caimans. He had a Gila monster that the story was that a friend had it, found it, got it. Don't know the story, out of Arizona, so it could be wild caught. And then he had these 2 rubber boas that came from Utah that were wild caught, and don't have a way to check to see if they were legally harvested or taken from the wild. And then he had a couple of live large snapping turtles, which are invasive species in Idaho, so we shouldn't have had those as pets anyway. Next slide. So there's the alligator up in the top. I will tell you that we gave that, he was not mean, but he's a big, strong animal. We gave him some drugs to slow him down, and then we used, 4 catch poles on him, and we still had to wrestle him hard to finally get him subdued. The Gila monster there was a nice full-size adult. The caimans ended up going to the Idaho Falls Aquarium, so you can see them. The rubber boas might have gone there too. I don't remember where they ended up. The Gila monster is...
I think that the rubber boas ended up in Zoo, Idaho, at least one of them.
Okay. And then the Gila monster is with Northwest Nazarene University. So he found a home, she found a home. I don't know what it is. And I don't remember what we did with the snapping turtles. We might have just given them to Department of Ag and said, here, happy birthday. Do I have another slide?
Nope.
Okay, so go back to my picture there. And the only thing I will add is my very first kid that I talked about, since we did the search warrant, I heard that he had gone out with another collector and collected 35 rattlesnakes, got bit, ended up in the hospital. I can't verify any of that. It's just a rumor going around. But what I would ask for this group, that when you're out and about, doing your work on, just out enjoying yourself, if you see somebody collecting or what you think is someone collecting herps, take a picture of the vehicle. If you can get a license plate, perfect. If not, just give me a description and a location of where that was, and we'll kind of keep an eye on who's doing what out on the landscape. We're finding, especially here, Boise, with our population exploding. We're getting a lot of rattlesnakes that are just showing up at dens dead because people are just shooting them. And then the more I keep my ears open, I'm finding more and more folks that are collecting rattlesnakes. There's a group here in the Boise area that is trying to change Idaho import rules to allow the import of any, how do they word it? Any cold-blooded reptile, any cold-blooded animal without an import permit. So if you can call it a pet, they want it to be able to come into Idaho. This is the second year, I think, that it's trying to get passed. So something to keep your eyes and ears open for. And then the last part is I'm seeing some discussions by some of the herp community catching these wild rattlesnakes and gopher snakes, that some of them are showing what they think might be snake fungal disease. They've got some scales that have some ugly looks to them. But for the most part, what I'm seeing in that community is after these snakes have a couple sheds, they clear up. So I haven't been able to get my hands on any of them. I do have 6 rattlesnakes in my freezer at the moment that I took away from one of these folks that we had to euthanize. I haven't had it tested or anything, but it's what I know. That's all I got, Chuck.
Okay, well, thanks very much, Charlie. That's always very interesting to see what's going on. Rita, are you prepared to talk about the State Wildlife Action Plan update? I see Rita on my screen, but I don't know if she's actually there yet or not. Okay, so we'll go ahead to Austin Wright. I mentioned this at our last PARC meeting, that the Idaho Master Naturalist Program that Austin is the author, along with John Cassell, of the herpetology chapter for the Idaho Master Naturalist Program curriculum. And I've got, I think, about 3 slides here. Austin, are you here? Would you care to take over and talk about these slides?
Yeah, I can do that. Can you hear me okay?
Yes.
All right, awesome. Yeah, that's me in the pictures. The one with the rattlesnake actually was when, as a student at NNU, I helped for a field trip, Christina Parker, do some rattlesnake studies. So, but basically the whole premise here is that I'm not new to the herp scene, but knowledgeable at least. And so currently I'm serving an AmeriCorps term, which is like the Peace Corps, but within the United States, at the MK Nature Center, which is a part of Fish and Game. And my supervisor there is Sarah Foch, and she does the entire, or she helps run all the Idaho Master Naturalist volunteers. And she was like, I have been looking for someone to write my herpetology chapter. I was like, well, what a coincidence. That's my background. So although my background actually isn't in specifically Idaho herps, if you want to go to the next slide, Chuck, that would be great.
Oh, hold on. There we go.
It's all in tropical anurans. So I just thought I would show you that's my background. So with Dr. Cassell at NNU. And I just like to point out, this is always fun. The booger that's on my finger in that picture is actually a full adult male frog. So that's just fun to point out. Next slide, please. So this is the chapter. Those are the kind of the goals. This is, I think, publicly available. So if you are in need of educating a group of people very, very basically about herpetology, I don't go into depth about research studies or research projects or detailed studies. My goal is to give a real broad scope. You know, even now, most people, when they hear me say herpetology, they think I'm talking about some sort of disease, and not reptiles and amphibians. So this chapter, it's not especially long, should be a broad scope document in which you can educate yourself on herpetology, as well as every species of reptile and amphibian here in Idaho, and a little blurb about that. Yeah, if you find yourself reading it, that's cool. And if you have any comments, you can send them my way. I didn't put my email in here. I'll put my email in the chat if you do have comments. But if you want to give it a read.
And you also are giving presentations for the on herpetology for the Idaho Master Naturalist chapter in Boise. Is that correct?
Yeah, the sagebrush steppe chapter in Boise. I'm the one who's giving the lecture for the Master Naturalists virtually again this year. Yeah, trying to help the scaly and slimy get a little bit more known in the area. Yeah. Anything else, Chuck?
No, I think that was it. But I thought that this is going to be a document that probably quite a few people are going to be using, and I thought it'd be nice to have you tell us a little bit about it. So thanks.
Yeah. That's all I've got.
Okay, great. I thought I'd give a brief update on the Idaho Amphibian and Reptile iNaturalist project. We started this 4.5 years ago. And I have not done a lot with it over the past year. It's within the project, there's about 3,562 observations. And then there's another 1,200 observations that I have not reviewed and looked over or added into this. But that suggests that even despite the pandemic here, that it continued to grow at about the rate that it had the previous year. Overall, iNaturalist has really taken off. When we started our project in 2016, there were about 1.5 million observations in there, and there's almost 60 million observations of over 300,000 species worldwide. But this is turning out to be a good source of information. Also, we use this along with museum records and the Idaho Department of Fish and Game road mortality database and the literature to put together a manuscript on road mortality in Idaho amphibians and reptiles. And that's been submitted. Julie Meredith is the senior author on that, an undergraduate student here at ISU. And that's been submitted to herpetological conservation and biology. So hopefully that'll be accepted. Couple of the major findings from that study are, one, we haven't documented road mortality and herps very well at all in the state. And secondly, there's more gopher snakes that have been, they make up like over 40%, or maybe it's 50% of the road mortality. So they're a species that's been, accounts for a lot of the road mortality. There's a reptile enthusiast here in Pocatello named Joel Reap, and he has really seen a lot of interesting uh, animals or species in the Pocatello area, including a desert night snake. That's the first observation for one of those since 1962. And then also we get more observations of ringneck snakes, but he found a road-killed ringneck snake this year. There was another observation of northern leopard frogs down around at Castle Rock State Park, where they had put in a fishing pond. And so that suggests that that species still is occurring in that area, even though there hadn't been other observations. There's one other observation a year ago, and then you'd have to go back 30, 40 years before that to see any. And I also just noticed when I was scanning through this that we're seeing a number of non-native turtles showing up as observations, particularly sliders, but also snapping turtles and things. And that's something that, you know, is happening all over the country, that turtles are being, you know, moved around, and non-natives are being introduced to areas. Okay. Rita, are you online yet?
Hey, Chuck, can you hear me?
Yes.
Okay, I apologize. Sorry. I'm dealing with an urgent critical matter that just popped up at work right as our meeting started, which is why I haven't been on. I was just typing you a message. So I've got to deal with something with our director's office, and I'll send a note in the chat box when I can get back on the meeting, okay?
Okay, no problem. Thank you very
Sorry.
much.
Okay, thanks.
Okay, here's another kind of statewide project that is going on, but I don't have any updated information on. But Josh Parker, who's a colleague of mine from California, Steve Mackesy from University of Northern Colorado, Todd Casto from University of Texas, Arlington. These guys are doing a NSF-funded study on Western rattlesnake hybrid zones. And they have been collecting all throughout the Western United States. And Josh Parker has been doing a lot of the field work for them. And so what was in 2019, he spent a good part of the summer getting specimens from Idaho. I know we got some from a den up in Lewiston and from out on the birds of prey area, and Patty Murphy from Fish and Game up in Salmon, and I helped him get some prairie rattlesnakes from up in the salmon region. This is a very well-funded, ambitious study. They're doing morphological work on these, but the exciting part is the genetics. And they're basically sequencing the whole genome for each of, I think, 20 individuals from here. And then they're going to be looking at, and they're doing that, you know, throughout the Western US here. And they're going to be looking at the what's going on with the systematics, but it's probably a far more complicated story than what we're seeing here with just naming. These are currently the Western rattlesnakes. And this all 20 years ago, these all used to be considered Western rattlesnakes or prairie rattlesnakes. That got broken up and prairies were separated out about 20 years ago. And then there's some people that think that the Northern Pacifics are separate from the Great Basin rattlesnakes. And I think what they're going to find is that the picture is more complicated, not as clear cut as that. But I have not gotten an update. I know that they were planning to do more work in Idaho last year, and because of the pandemic, that was scrapped. So anyway, this is a project that we want to keep an eye on because it's going to inform us about the actual identity of our rattlesnake populations. Okay, well, I suggest that we take a break here for about, let's take a break for about 10 minutes. Now, I have a thing that I've been using with my classes here, and this is a program called Mentimeter. And if you'll go with some device you have, the computer or a cell phone or an iPad, go to www.menti.com, and then it'll ask you for a code. Put this code in, and then you can sign in, and this will, then I'll have a record here of everybody that's been able to take part in the meeting. So I'll see you back here at about 5 after 3 Mountain Time. Okay, I think we'll try to get started again. So the way I organized the rest of this is that we would go through by region, and we're fortunate to have Michael Lucid to talk to us about what's going on up in northern Idaho. He's recently published a paper in Herpological Conservation and Biology on the status of amphibians in northern Idaho and eastern Washington. And I think he has a few other things to tell us about too. Are you ready to share your screen, Michael?
Yeah, I'm ready.
Okay, let me give you, well, I hope we can, let's see. Let's see, I'll stop sharing and hopefully it'll let me allow you to share. Okay, you should be able to share now.
All right, can you see that?
Yes.
All right. Is it a PowerPoint or is it the weather forecast?
I'm seeing a PowerPoint.
Oh, good.
You're in the, oh, I forget what it is. It's not, there you go.
All right, perfect. Is it like the full screen?
Yeah, you're full
Okay,
screen now.
sweet.
Great.
All right, awesome. Yeah, cool. Yeah, thanks for inviting me to join. Yeah, for those of you that don't know me, I was Fish and Game's diversity biologist up in the Panhandle for about 10 years until last year when I left the agency to start a consulting company called Selkirk Wildlife Science. And what I'm talking about today, we got a couple papers coming out and we're still kind of thinking around with trying to publish the data we collected over the last 10 years. And Chuck asked me to talk about a couple papers we have out now and what's coming next. The first one is a paper that came out in December in HerpCon Bio, and it was just a summary of the dipnet larval surveys we did across the Panhandle and in a little bit of northeastern Washington in 2013. We did dipnet larval surveys at 433 ponds, and the goal of that was to just assess the status of native pond breeding amphibians. And this is what we found. These are just pictures from the paper and you can download it. It's open access if you want to look at this in more detail. But if you look at Columbia spotted frogs, were our most commonly detected species. They were pretty ubiquitous across the landscape. And we did one survey in each of these 5x5 kilometer cells. Long-toed salamanders were also very common, slightly less common than Columbia spotted frogs with our detections. Syren tree frogs were a little less commonly detected than I really expected them to be, but we kind of had a little bit of a dearth of detections in the Coeur d'Alene Mountains here and in the eastern part of the St. Joe. Bullfrogs, I'll skip to them, were pretty common, unfortunately, but mostly in the valley bottoms and the lower elevation areas. And western toads were kind of few and far between. We only found them at 16. We only found them breeding at 16 different sites in 2013. And then the other side of the story was there's two other species who are thought to be native to northern Idaho. One's northern leopard frog and the other one is wood frog. And we did not detect either of those species. And so we set out upon a little mission to figure out if those species were actually native to northern Idaho. And the way we went about that was we looked in the literature and we acquired every museum specimen that we could find. This is a museum specimen of a northern leopard frog. And by looking at the literature and getting museum specimens, we were able to ascertain that northern leopard frogs are indeed native to the Idaho Panhandle. Most of the museum records are from the northern part of the Panhandle. And the most recent verified detection of a northern leopard frog in the Panhandle of Idaho is 1955. And I believe this is that specimen. And they went as the museum specimens go as far south as Lake Cocolala, which is kind of halfway between Coeur d'Alene and Sandpoint. And so our conclusion was that northern leopard frogs are most likely extirpated from the panhandle of Idaho because we didn't find them and we were able to verify that they're a native species. There is a native colony that's extant just north of the panhandle at the Creston Wildlife Management Area in British Columbia. And the other one was the more exciting story. For years and years, there's been this kind of natural history mystery that there's some mystery population of wood frogs way up in northern Idaho. And this all started in the 1940s. There was a zoologist by the last name of Dumas who published a couple of short notes saying that he found wood frogs here and there in the panhandle. And then in the 70s, there was a big collection up in the Priest Lake area of specimens. And then there were several other specimens. And we looked through the literature and the descriptions of the species, and then we obtained the specimens, and they're quite obviously spotted frogs for all the characteristics that are pointed out on this slide. There was one museum specimen that we were unable to find. It was I don't remember, Chuck, if it was at ISU or U of I, but Chuck helped me try to find it, and it is AWOL. It was unable to be found. So unless that particular one happens to be a wood frog, we found no evidence supporting that wood frog were ever a native species or there was a population extant anywhere in Idaho. And then the second paper that we have coming out is called Genetic Structure Not Detected in Northern Idaho and Northeast Washington Western Toad Populations. We collected genetic samples during that same survey. And this was kind of driven by both Idaho and Washington State Wildlife Action Plans. There was a conservation action to determine the taxonomic status of Western toads, because there's been a lot of cryptic diversity detected within that species group. And so we took tissue samples from toads that we collected in the field, and we worked with Jack Sullivan at the U of I to sequence a very short section of the CO1 gene. And we produced this very complicated figure that I'm sorry to have to share to you, but I can't tell you how many edits I had to go through with the journal editors to let them let me publish this. But here it is. It's complicated, but basically the story is we compared our short section of CO1 with a previous study that was done in 2009 of Western toads across the Western United States, across their entire range. And that was by a person named Goebel. And she basically came up with 3 major clades. And one of them is that big one on top, the northwestern clades. And all of our toads fell within the northwestern clade of Western toads, which is exactly where we expected them to. And also, interestingly, we had no real structure within our study area. And, you know, as you can see, we had a big area, and even this isolated population out in the St. Joe was, it was mixing in with animals from the Cabinet Mountains, from the Selkirk Mountains. And so we didn't identify any obvious structure within our study area. But the caveat is it was a very short section of CO1. Goebel used 3 different genes and did a much more complex analysis. But I think we reached our goal as to being able to say that the toads in our study area are appropriately assigned to Anexurus boreas. So, and that was all done in 2013. In 2018, we, when I was still working for Fish and Game, and Casey McCormick, who is the new diversity biologist, and I think I saw his name that he's on here. He was the technician in the region at the time. We set out to resurvey as many of these ponds as we could 5 years later. And we managed to pull off resurveying 123 of them. So we have 2 surveys with 5 years between them of the same sites, same methods. And what we found was, if you look at the bottom, all of our native species, and this is just breeding detected, not species detected, we actually found the exact same number of sites. There were some changes to the actual site. So we had a 0% change in where we were finding native amphibians. I'm just gonna work my way up from the bottom to the top. Bullfrogs, we found at 5 sites in 2013, 5 in 2018, but that's just kind of an artifact of we didn't prioritize surveying bullfrog sites. There's lots of bullfrogs out there. And then tree frogs were about the same, 12 sites in 2013, 15 in 2018. Spotted frogs, we actually found an increase in breeding by about 23% from 47 to 58 sites. Salamanders, long-toed salamanders, we had almost the exact same number of sites, but they were really interesting because they actually really switched sites. We only found them at about half the sites we'd found them at in 2013, but we found them at 24 new sites in 2018. And then the big story from, I ruined the surprise. So it's not a good surprise, but Western toads were kind of the big story. We had only 16 sites where we detected breeding in 2013, and we had only 8 sites in 2018. And so what you see here is the map where we detected toads in 2013 only has a big red cross through it. The ones where we detected them both years are in black, and then we only had 1 new site. And remember, we surveyed 123 ponds, and we prioritized making sure we resurveyed every site where we detected toads in 2013. And so we see about a 50% net loss of sites where we're detecting Western toads in our 2 surveys. And of course, it's only 2 surveys, but 2 surveys is more than we have for most big amphibian projects. And this is really matching up to what I'm seeing out on the ground and other sites beyond our survey. I'm just not seeing Western toads like I used to see them. And there's sites where I used to see tadpoles every year, and I'm just not seeing them anymore. So I am fairly concerned about Western toad populations in the Panhandle. And I'm working with a couple of colleagues to try to, at a minimum, just get this in the literature, even though we can't say anything for sure with just 2 surveys as at least a note. And been working on trying to find some funding to do something more sophisticated. I would really like to be able to provide some insight as to why this is happening, if it is indeed a big population crash, and try to figure out what's going on. So if anybody knows any good funding sources for such work, feel free to reach out and let me know. That's the last slide we got. This is our company website. We're just selkirkwildlife.com. We have a real wide range of expertise in taxonomy and techniques. And this is just my business pitch. If anybody has projects and they'd like to have help with, feel free to reach out and talk to us. And that's all I got.
Okay, well, thank you very much. So let's see if you can stop sharing.
I will try.
And then I'll go back. Well, oh, there we go. All right, is that working? Okay, let's see if I think I can share now. Okay. So thanks a lot, Michael. That's most of the sites with Western toads in southeast Idaho below the Snake River have pretty much disappeared. There's only a few where they are still persisting in the southeast, but it seems like with Western toads, it's things are different in different places, that they seem to be doing well in some places and not well in the others. I remember we were worried about them 20 years ago up in the Panhandle, and then there were quite a few observations that came in, but now it sounds like there's maybe something going on again.
Yeah, it sure seems like it. I mean, like I said, with 2 surveys, there's not a whole lot definitive we can say, but it sure is worrying to see it out on the ground and where you're just not seeing them where you used to anymore, and to have that data to go along with it.
Yeah. They're an interesting species too. They seem to be disturbance related, that we can go a while without seeing breeding, and then there's a forest fire or some kind of disturbance, and then you'll get a pulse of breeding. So it's I think a challenging species to figure out. Did you do any swabbing for BD or ranavirus?
Yeah, we did. For the most part, we focused on spotted frogs for that, just because the goal was to kind of get distribution across the study area, and we didn't anticipate detecting a lot of toads.
Mm-hmm.
And we did find it's pretty ubiquitous. We detected BD at 66% of the sites where we surveyed. And we are working with Jennifer Wack, who is a professor at Eastern Washington, and she's a microbiologist and skin fungal specialist. And she has a master's student that just finished up working on some of our data, and he is working on a publication from what all he learned. I'm not sure exactly when he's going to finish that up, but we didn't do much that was focused on toad swabs, but that would be good to do.
Yeah, I remember he presented at the meetings a year ago, if it was the same fellow.
Yeah, Philip.
Yeah. Well, David Pilliod and a number of other biologists were studying a large population of western toads just east of Grand Teton National Park. And I mean, it was a big population, hundreds and hundreds of toads. And yet they found that they did the swabs and they did mark recapture, and they found that the ones that had the tested positive for the BD had lower survivorship. And so they at least at that point in time, I don't know what's going on now, but then they thought that the population was in slow decline. It wasn't a huge population die-off, but the population was declining, even though it was a big population. So, okay. I don't have any information on any herpetological work in the Clearwater, other than in 2019 there were those rattlesnakes collected. Does anyone from there have anything to add about the Clearwater region? If not, we have an interesting observation from the McCall area. I think this was at Ponderosa State Park. This was in December. Diane Evans-Mack, who's the diversity biologist in the McCall subregion, they were out doing their Christmas bird count, and a long-toed salamander came walking across the ski trail. And you can see him here on the snow, and then in someone's hand. And I've heard about them in the springtime, making their way over the snow to get to the breeding ponds, but I hadn't heard of, I didn't know of any observations occurring in December, but I thought this was a really, really neat observation. Okay, for the Southwest region, Christina Parker, who's a graduate student at Boise State University, working with Jay Carlisle and David Pilliod. She is going to tell us a bit about some of the work that she's been involved with there. So are you ready, Christina?
Yep, I'm ready.
Great.
So I will start out with talking about this paper that I was fortunate to participate in, and other people that are here at this meeting also participated. But we were able to look through this paper on illegal shooting of non-game in the Birds of Prey National Conservation Area, that it appears that rattlesnakes are being targeted versus kind of an opportunistic shooting. And this can be a big issue because the public being aware of the few hibernacula and targeting them at these locations. And you can see in this table down below that many of these incidences where we've come up to hibernacula or just high areas of rattlesnakes, that there are gunshots and casings that are being left behind. I'm going to go to the next slide. So for my thesis, I was able to survey inside the Birds of Prey area for 2 years. In 2018, we observed 7 snake and 7 lizard species out of a total of 16 reptile species that could be at the NCA. We see that we have a lot of observations of the side blotch lizard and whiptail lizards, and lots of observations of gopher snakes, which typically we assume to be most common snake out there in the NCA. You can go to the next slide. Back one. There. In 2019, we did a different survey method. 2018 was trapping. 2019, we did visual encounter surveying, and we detected 4 snakes and 6 lizard species out of a total of 16 possible reptiles. And by doing the visual encounter surveying, we still saw lots of lizards, primarily side blotch lizards and whiptail lizards. One surprising thing about 2019, although it was a different survey method, we didn't see a lot of gopher snakes, which is a little bit troubling and unsettling because we did do some snake fungal disease swabbings and found that we do have snake fungal disease on gopher snakes. So that's one thing that we're thinking about that might be relevant to why we see a decrease in trapping.
It's a different observation method, Christina.
Pardon? Sorry, I didn't hear the question. Okay, well, Chuck, you can go to the next slide. Through live trapping, we were able to detect 2 of like the 3 what we call like rare snake species here in Idaho, the desert night snake and the ground snake. Both of these, last time that they were detected through trapping was in 1998. So it's a big time difference between the 2. And then we didn't detect any long-nosed snakes, but last time those were detected was in 2012. This coming field season in 2021, we're going to be researching to go to historical locations where we've detected these rare snakes, either by trapping or using citizen science data, and trying to detect and get a better inventory of what these species, where they're at, and if we're still seeing them in their range. Another thing that we're doing this field season will be swabbing for snake fungal disease at any opportunistic snake that we encounter. And I also know that the Idaho Army National Guard will also be swabbing for snake fungal disease this summer as well. So that's kind of an update and a wrap up of what's happening in the Southwest area.
Sorry, I was, Christina, I was asking, is there anything more to add from what we had last year on snake fungal disease?
Not to my knowledge. I wasn't out very much in 2020. So as far as any new updates on snake fungal disease, I don't have any.
Uh-huh. Well, I know, Charlie, you said that you saw some, you had some photographs of snakes that looked like they had some problems with their scales.
Yeah, it's social media type stuff, guys sharing back and forth that I've seen, but I haven't actually got to see anything in person or have a photo sent to me.
Uh-huh. Okay. Well, thank you, Christina. Okay, and now we have we're also fortunate to have Tempe Reagan talking to us about work that she's been doing for the past couple of years on surveys for alpine species of greatest conservation need in the salmon region.
Yeah, thanks, Chuck. On some of these slides, there's going to be animations. So I guess if you see blanks or something, click, or I'll just tell you to click when it's time for that stuff to come in.
Okay.
Let me minimize this talking head here. Yeah, so I'll just cover briefly the alpine surveys and amphibians as it pertains to those, and then delve into our amphibian work in Region 7, and then talk about the statewide efforts that Idaho Fish and Game is conducting. So as far as the alpine surveys, we initiated a multi-species alpine survey protocol in the salmon region in 2019 for probably 10 species of greatest conservation need. And part of the protocol is during those surveys, we conduct opportunistic visual encounter surveys if amphibian habitat is present. And we conducted a second season in 2020 in the salmon region, as well as expanding this protocol statewide. And we plan to continue these baseline surveys for the next 2 years statewide before kind of moving on to more specific or different questions. Next slide.
Nope. Is that correct?
Yeah, yep.
Okay.
Oh, the back previous one. Yeah, okay. So this is the 2020 overall results for the salmon region. We conducted a total of 37 of these alpine surveys, and the map shows in yellow the survey sites we completed alpine surveys in. And these are based off of a spatially balanced random sample of 4x4 kilometer grid cells. And then within those grid cells, we select an alpine basin that's easy to access, easy being the operative word here, and then backpack in to conduct the surveys. We were able to cover 6 different mountain ranges within the salmon region and spent a total of 107 days conducting those surveys. I mentioned the other regions were on board in 2020 conducting surveys, and I don't know the exact number of surveys they conducted, but probably close to 20 other surveys were conducted statewide for the first season. But as it pertains to amphibians, you can go to the next slide. So in 2020 versus 2019, amphibians were basically the same, although in 2020, we redid the sample draw. So we were generally surveying new sites. We detected the 3 common species you would expect in 2019, western toad, Columbia spotted frog, and long-toed salamanders during 7 different surveys. Sorry, my bird might chime in during this. In 2020, again, we detected those same 3 species at 7 different survey sites. But the really unique thing that I will preface with a caveat, we're not 100% sure, we're going to verify in 2021. My technician at 2 spatially disparate sites was finding salamander larvae that were extremely large, like beyond anything I had ever seen in a long-toed salamander, and with potentially different gill design. And I kind of consulted with David Pilliard via pictures because I was suspicious that they could be tiger salamanders, which have never been recorded, nor is the salmon range part of their historic range that we know of. And he thought that they might be tiger salamanders and posed the question whether those are populations that have been introduced by fishermen to those lakes as bait, or if those are remnant relic populations from many, many years ago. So we're going to try to access both those sites in 2021 and collect genetic samples. My technician was never able to catch a live one. They were too fast. He did find a dead one and did not bring it into the office because it was like moldy and nasty, but I have pictures of that one too. So that's pending potentially very interesting result of these alpine surveys. So
yeah, that's not, that doesn't seem to me terribly unlikely that those could be tiger salamanders, because east of there in the Targhee National Forest, you have tiger salamanders.
Yeah, and the one thing that's interesting too is one of those sites is a site that doesn't have a trail to it. It's extremely remote. I don't think it has fish in the lake, so kind of doesn't lend itself to the argument of fishermen introducing these tiger salamanders as bait or escaped bait. So we shall see. We could be totally wrong. They could just be gargantuan long-toed salamanders, and we will find out, and I'll update you next year.
Yeah, this is really interesting to me. I'm excited to hear what you find out.
Yeah. Next slide. So kind of moving on to an overview of Idaho Fish and Game, amphibian, and wetland monitoring. So again, this started out in the salmon region in 2019. The map shows the salmon region highlighted there. And the basic protocol we derived from a previous study conducted in 2012 and 2014 in a BLM cooperative study with Idaho Department of Fish and Game, and that was conducted in regions 5, 6, and 7. So in 2019, I was interested in revisiting sites that they had surveyed in 2012 to determine turnover of wetlands, whether they're wet or dry, and if the same species are holding strong, or if we're seeing new species come online and other species disappear. And then we were able to expand with the rest of the wildlife diversity program in 2020. These surveys, we went statewide. Michael Lucid and I actually worked together to develop this study prior to him starting his consulting company before he left Idaho Fish and Game. And so in 2020, I'll just make a note that we really were interested in focusing on western toads with these statewide surveys. And so what Michael and I did was go through all of the historic western toad data in our Idaho species database. And we defined, I think, 345 historic western toad breeding sites to be hopefully resurveyed in the coming years to determine if toads are still present. So next slide. So yeah, the methodology was basically derived from that 2012 study I mentioned using visual encounter surveys. During those surveys, we also collected a suite of like very detailed wetland habitat characteristics. Hopefully, I know that the previous study, they were looking at actually calculating occupancy. I don't know if they did that, but that's what these covariates could actually be used for in the future. In 2019, we also conducted Kitchard surveillance at 20 sites. In 2020, we kind of tailored the Kitchard surveillance toward western toads. And so we were only collecting Kitchard swabs from adult western toads in 2020. Next slide.
I thought I'd just point out that it might be worth your while to measure conductivity as well. because at least in Yellowstone, we find that the toad breeding is strongly correlated with high conductivity levels. So that might be, and there's some suggestions that the high conductivity water is associated with their ability to deal with BD as well.
Yeah, okay, thank you. All right, next slide. So I'll go over the results for 2019 for the salmon region and then the 2020 results statewide in the salmon region. So for 2019, we were able to survey a total of 133 wetland sites. In those, we revisited 74 of the sites that were initially surveyed in 2012, and we added 59 new sites. These sites are sites that we would observe in the field conducting the other surveys and then realized they had never been surveyed before, and so we would just add them to the survey suite. So the map shows just the dispersion of all the sites surveyed in 2019, and you can click Chuck. So we detected amphibians at 48 of those 133 sites. This map that just popped up in blue shows the dispersion of positive amphibian sites. Similar to what Michael mentioned, the most common species we observe in the salmon region are Columbia spotted frogs. Next, you can click again. Of the sites surveyed, 22 were dry. So 16.5% of the sites visited, whether they most of these were actually historic sites that we were revisiting, and we found that they were now dry. Okay, you can click again. I think, okay. So this is kind of just like turnover in sites that of the 74 sites we were able to revisit. So positive to negative, that bar indicates a 17.6% change in sites that were positive for amphibians in 2012 that we then found were negative, or no amphibians were detected in 2019. It doesn't necessarily mean amphibians weren't there. It just means that we did not detect them on our surveys. Because this is basically presence absence, we didn't actually calculate occupancy with our data. So we can't say any more than that. Turnover for sites moving from negative to positive, 10.8% of sites that were initially negative in 2012 now had amphibians or had amphibians detected in 2019. And then the percent change of wetlands from wet to dry was 6.8%. Next slide. Okay, so then the Chytrid results were actually quite interesting, I thought. We swabbed 60 individual amphibians for Chytrid fungus from 21 different wetlands in 2019. Of all of those individual swabs, 44 out of 60 tested positive, so 73%. 12 tested negative, 20%, and 5 samples they were unable to actually determine the status. So 18 of our 21 sites actually were positive for Chytrid in 2019, and only 3 sites were fully negative. And what I mean by fully negative is that all 3 swabs that we ran tests on tested negative. And the map shows in the different colors the status of sites. So the yellow stars, which you can really only see one, is a site that had Chytrid present in 2012. Black stars indicate sites resurveyed for Chytrid. 2019 positive Chytrid sites are green and Chytrid absent sites are purple. And you can click again, Chuck. So in 2012, what they found was that only 2 of 51 sampled individual amphibians tested positive for BD, and only 2 sites were positive for BD in the salmon region. You can click again. All sites that we resurveyed for Chytrid in 2019 from 2012 were positive for Chytrid. You can click again. So of all the sites that they tested for Chytrid in 2012, just 0.09% were positive versus 86% of our sites sampled for Chytrid in 2019 were positive. So that was pretty striking to me, and kind of was impetus, at least for me in my region, to continue Chytrid surveillance and or think about ways that we can, because you can't cure Chytrid. So what are ways that we can buffer our amphibian populations to make them more resilient to the fungus? But that's in the works. Next slide. So this is the 2020 results. I haven't had time to make all the figures because I've been spending much time making alpine figures. So in 2020, it was really awesome. The rest of the state got on board and we collectively surveyed 142 sites statewide for amphibians. 100 sites were in the salmon region and 42 were across the rest of the state. I mentioned that the focus in 2020 was geared towards Western toads, and we started with a potential survey option of 345 historic sites. The goal being, can we eventually revisit all of these over time? And so in 2020, we surveyed 62 of the 345 sites. Let's see, okay, yeah. Toads were detected at 28 of those 62 sites statewide, so 45% of the sites had Western toads still, plus one site in Region 7 that was not originally classified as a historic Western toad breeding site. I mentioned we only targeted fully formed Western toads for chytrid sampling in 2020. We also took genetic samples from larval amphibians of all species encountered, and analysis on those samples is pending. But just to go through the chytrid, again in Region 7, so because we're only targeting adult Western toads, we have many less samples because you don't find adult Western toads that often, but we collected 24 swabs from 7 different sites. 18 of the 24 swabs were positive for chytrid, and 6 of 7 sites sampled were fully positive for chytrid. Only 1 was fully negative, meaning all 3 samples collected tested negative. In Region 4, so that's the Magic Valley region, they collected 3 swabs from 3 individual Western toads. All were negative. In Region 5, 1 swab was collected, it was positive, and we're still awaiting our results on 9 swabs collected in Region 6. Next slide. Oh, again, most common species detected statewide was Columbia spotted frogs, and I will mention there was 1 detection of northern leopard frogs in Region 6, and we're not targeting northern leopard frogs at the moment, but that is something that some of the biologists were talking amongst ourselves about, northern leopard frogs, because there is a suite of sites that had positive northern leopard frog detections in 2012 during that larger study. And so it might be worth just re-verifying if those frogs are still present at those sites, especially in Regions 5 and 6. I don't have northern leopard frogs in Region 7, unfortunately. So anyway, if there's any questions, and if there's time, I'm happy to take questions, but that's just a little update on what we're doing. We are planning on continuing these surveys through at least 2021, focusing on Western toads, and might delve a little bit more into trying to get some statistical estimates on occupancy and detection for Western toads, at least in the salmon region. I'm not sure about the rest of the state yet, so, but we'll continue to focus on toads.
Great. So, yeah, thanks very much for that. That's very interesting, kind of disturbing.
Yeah,
what's happening there. It seems that the higher elevation toad populations are having more trouble too. And I know in some cases this is related to temperature and their ability to clear the chytrid fungus if it's cold. Yeah. Well, I think that's terrific. You guys are doing that. I'm really happy to see that you're looking at that. It's frustrating though, because you find that, and what do you do?
Right. Yeah, at least in the salmon region, what we've noticed is like the biggest disturbance or negative impact to our wetlands, which mostly occur on lower elevation public land, is free range livestock grazing. So if they're, it's just in the future, can we work with land management agencies to really work on erecting and maintaining exclosures on those wetlands? Hopefully, if the quality isn't as degraded, the individual amphibians themselves will be more resilient and able to overcome BD infection.
Yeah, the use of exclosures with toads, you've got to be careful about that too, because if the vegetation grows up too much,
right,
it's not good for them that way either. there was a study done in Great Britain with the toad that they have there, and they put up exclosures, and they found they had to take them down because they were actually inhibiting the breeding. So it's not maybe as straightforward as you might sometimes think.
Right. And I will mention to that point, unfortunately, most of the toads, most of the sites that have toads in Region 7 are not as low elevation. I don't know what that means, but they're usually kind of restricted to higher elevation areas. Or areas that, yeah, maybe are more protected. I don't know.
Uh-huh. Okay, well, thank you very much. Are there any questions from anyone else?
Yeah, I have a question. So I know that with fisheries, they've been opening up a lot of those lower parts in your region of barriers that were prohibiting some on migrations, especially in the Pacific tributaries and stuff, because I was involved in doing the disease work above and below and fish. I think opening up some of those areas have caused migration of some of the amphibians so that the BD is spreading. Because that wasn't something that we were looking at when we were looking at opening this up. We were looking specifically at fish pathogens.
Was that a comment or a question? I think
that's a question.
Comment.
Question.
Oh.
Do you think they're related? Because I know we opened 17. I know at least 7. There were 17 slated to be reopened.
In the Pacific or region wide?
Region wide.
You know, maybe that's part of it. The Pacific, we did quite a few amphibian surveys there, but a lot of them didn't detect amphibians at all. A part of me wonder, I don't know why it spread, or, you know, part of me wonders if recreationists are spreading it or if livestock are spreading it. I mean, it spreads the spore. So I imagine there's like 100 different vectors that can easily transmit it.
Well, I know Jody Brostrom was working on a lot of those projects down there, as well as Mike. So I know she's retired now, so I can't talk to her as well.
Yeah.
I used to check with her and find out, because I know that a lot of those sites, you know, we actually just came out, Southeast PARC actually just came up with a heavy equipment decontamination protocol for prohibiting, you know, specifically looking at BD. And fish pathogens are included in that because they're recommending Burcon. But with that, I don't think a lot of that equipment, when I know that they were looking at that before, and there wasn't really anything they could do when they started opening those things up. And so if people are, you know, tromping around in there, I mean, it's a possibility, but it is curious.
Mm-hmm.
Okay, well, thanks again, Christina or Tempe, and we'll proceed on here. Does anyone know about, if anything, well, I guess Tempe told us a little bit about the what was going on in Magic Valley, that they had done some toad swabbing there. Anything else that anyone knows about from that part of the state?
Hi, Chuck. Can you hear me?
Yeah.
This is Lynn Snoddy. I'm the diversity biologist in the Magic Valley, and yeah, there's nothing really to report other than the few amphibian surveys I got to do last year. We'll plan on doing more this year and hopefully have more to report next year.
Okay, great. All right. For Upper Snake, there's a continuation of the at the Camas National Wildlife Refuge of the cover boards study, and that is coordinated and led by Alan Merritt out of Idaho Falls. And this kind of goes back, I think Alan was involved with the amphibian surveys there back in what, 2012 or 2013. And then I think it was 2015, my lab was asked to help out with some reptile surveys up at Camas National Wildlife Refuge, and we didn't find very much. And we had thought that maybe cover boards might be a good way to look at that. And so Alan put together a team. And what, you've been doing this 3 years now, Alan?
Yes, sir. The cover boards, 18, 19, and 20.
Yeah. So feel free to add in whatever you like here. I think that most of the species have been detected either under the cover boards or going, seeing them. Like there's a leopard lizards, isolated population of leopard lizards out here. I think it's the northeasternmost edge, right in this area here, and they've seen those, but we're not seeing the except for I think sagebrush lizards. The sagebrush lizards have been using these quite thoroughly, but you don't find that many other reptiles using the cover boards. Is that correct?
Well, the little sagebrush lizards are the most numerous, most prolific in that area. We have found at one time or another every other reptile that we know of is in the area. The only exception being we've never found a what we would consider a full-grown rattlesnake under one of these cover boards.
And you've been experimenting with different spacers under the boards to try to accommodate for that, right? Or allow the possibility of larger snakes to use them?
Yes.
And I know they detected skinks with these, or you detected skinks, which we hadn't seen in our surveys. So they've been effective for showing up some of the species, but it's required quite a bit of effort to get the things that you have. And you're thinking about expanding that this year to try in some other areas where the rattlesnakes are present, but they're further away from the rocky areas?
On the picture at the top that's got me lifting up the board, you can see a large kind of a rock pile ridge behind me. All of rocks in the foreground and all of that. And well, I think what we've concluded is that the adult snakes just come and go back into the rocks. You know, that seems like that's a preferred habitat, and they just sort of ignore the cover boards.
Mm-hmm. Well, I commend you guys for the amount of effort that you put in out there, and I've enjoyed coming out and seeing things. Here's a little juvenile rattlesnake. That was, I think, in September that we found that guy.
Yeah.
So, but anyway, we'll be interested to see what you come up with in this coming year.
Yeah, we've seen a couple of, they're very small, very brightly colored. We think they're recently hatched rattlesnakes. These little guys, 6 or 8 inches long. They're just gorgeous, but we never seen anything we would consider an adult.
Yeah, when we first started going out there, when I went out there with Sean Miller, we found several rattlesnakes in this rocky area. I think it was just a little bit back from here, but we haven't seen them since. We put out trail cameras there and weren't able to find them, and except for that one time, that's about the only time that we've been able to see them.
Well, I've been out there a few times myself, and if you get back in there and start walking around in the rocks, you'll hear them buzzing at you from inside the rock piles.
Yeah, this is right on the border of the National Wildlife Refuge and BLM land to the north there.
Yes.
Okay, well, thanks.
Thank you.
I have one other comment here on the regional review. And this has to do with the Blackfoot River restoration project. This is a large project. The Blackfoot River Wildlife Management Area is just east of Blackfoot Reservoir. It's up near the upper portion of the Blackfoot River. And I think it was about 25 years ago, or 25, 26 years ago, that this was acquired by the state, and particularly for trout habitat. And one of my graduate students back then, I think it was 1996, did amphibian survey. And then recently, I think it was 2 years ago, they started on doing a restoration project, trying to stabilize the river banks and to deepen the channels and to expand the wetland areas. And we thought that that would be a good chance, opportunity to look and see how things compared after 25 years. So the work has been done by an undergraduate student, Kenneth Long, and this is a comparison of what he found in 2020 compared to what Raymond Clark found in 1996. And he found fewer tiger salamander, this is number of sites, fewer tiger salamander sites, more places, quite a bit more with northern leopard frogs. I think that Raymond only found leopard frogs at one site, and there were quite a few more found by Kenneth. Raymond found more sites with chorus frogs back 25 years ago, and but still quite a few sites with chorus frogs now. And then these are the actual breeding sites. And then he saw a lot more terrestrial garter snakes, and Raymond never saw a common garter snake there, but Kenneth found one this year, but we found several when we were just starting to look around there a year ago. So for the most part, things just seem to be, stable or improving in that area. Here's just a map showing you what's going on there. The red are the 2020 observations. The blue are the 1996 observations. And they've done restoration work in this area last year and in this area this year, and then they'll be doing other areas, I think, next year as well, and maybe even beyond that. So it'll be interesting to see in the future, several years from now, repeat this and see if things have improved at all. But this is good news that the leopard frogs are, that there's more leopard frog areas there now. Okay, Rita, are you gonna be available to talk about the State Wildlife Action Plan? Okay, why don't we take a 5-minute break? We'll get back at 10 after. And if Rita's available, she'll tell us about the State Wildlife Action Plan, and then maybe we can discuss ideas for next year's park meeting. Or if anyone else has anything else they want to discuss, feel free to do that. The floor will be open. So let's say at 4:10, we'll start up again. Oh, for some of you may have joined since last time. If you didn't get a chance to sign in, it looks like there's about, a half dozen people at least that haven't signed in. You can go to www.menti.com, use this code and add your name and add your affiliation. There's going to be a gap in this, but I can hopefully that this worked in terms of the recording of the video, and I can post that on my YouTube channel if anybody wasn't able to be here for the whole thing, or you want to share it with someone else, we can do that. So if not, I hope maybe we'll interact later at other aspects of the meeting. I'll stick around for a few minutes if anybody has any questions or wants to talk about anything. But thank you all for attending and for the people that presented.
Thank you, Chuck. Thanks for organizing.
Oh, you're welcome. I enjoy doing this.
Hey, Chuck, can you link your YouTube channel in the chat real quick?
I'll see if I can. Well, I will. I've got different things in there, so I will. I'm going to have to create a playlist for Idaho PARC, but I'll send that to you, David.
Okay, thanks.
You bet.
Hey, Chuck, this is Paul Makala. Hey, I'm not sure how much feedback you're looking for right now, but as far as next year, any of those hands-on workshops, I think, are always really appreciated by the field staff. So whether you do reptiles or snakes or amphibians or what have you, I think that's a really good thing to continue.
Yeah. Oh, one thing I forgot to mention that might be of interest. I'd like to get people's response to this. I've been, working with the Park Service for a long time on the amphibian monitoring in Yellowstone, and Andy Ray, who's their ecologist in the Greater Yellowstone Network, and I are offering a course. It's a 1-hour seminar course. We meet at 7 o'clock on Tuesday evenings. It's virtual, but we're going through all of the amphibians of Yellowstone. And we've also gotten about a half dozen researchers to talk about disease and habitat fragmentation and landscape genetics and climate change. And so it's turning out to be a very interesting class. I was thinking that I could offer something like that in collaboration with the other universities and colleges in the state in the future. And I wonder how many people might be interested in taking that. Like, we wouldn't want to try to do all the species, but if we just did like snakes, or we just did salamanders or anurans or lizards, I think we could have a really interesting 1-hour seminar class that people could take. Do you think that would be of interest to your people, Paul?
Oh, absolutely. Yeah, you bet. I think that'd be a pretty exciting approach.
Well, and see, what's cool about that is that we can get various people with the virtual format. We can get, a variety of people to talk. So like in our Yellowstone course, we have the park disease biologist. We've got Melanie Murphy, who works on amphibian landscape genetics. She did her PhD in Yellowstone on toads and chorus frogs. And David Pilliot is talking about spotted frog movements, and Steve Spear is talking about tiger salamander landscape genetics. Paul Bartel's talking about habitat, temperature biophysical ecology. So we're teaching it. We're not having any prerequisites for it, and there's no real testing anything other than that people just participate in the class. But we've been having a lot of fun with it, and I think that might be something really fun to do in the future. What I'd like to do is collaborate with interested professors at other institutions, and then those courses could be offered for credit at other colleges and universities in the state. And then I've been talking with our people here at our Instructional Technology Resource Center, and we can set up a course so that we use our course management software and people can access it from elsewhere. But we do have 2 students from Bozeman taking the class, the Yellowstone class, but it ends up costing them about $400 or $500 to do it. And if we could collaborate among the universities, then people could probably just add it on without it costing them extra money. Does anybody else have any ideas for the meeting next year, or what you'd like to see? Charlie, perhaps something that would be useful is to actually go over all the legal restrictions and what's allowed and what's not allowed for collecting and keeping herps in the state. Okay, well, if there is no other comments or nobody has any questions and things, I think we'll end the meeting then now. So thanks for attending. See you later.
Thanks, Chuck.
Chuck?
Yeah.
At some point here in the near future, I was wanting to chat with you a little bit about leopard frogs if you had time. I don't know if we want to do that today or.
Well, I can chat with you right now if you like. I'm going to stop the recording here just a sec.
Okay.
You probably don't want to.